are entering into the next topic. It is coming under genetics. That is our human genome project. Okay, Cynthia?
Yes.
Okay. What is your idea about this topic? Human genome project? Hello.
Uh, to calculate, yes, ma'am. To calculate the number of genes
Mm.
present in the human body, base pairs determining.
Okay.
Uh,
Base pairs determination. Okay. So it's a project. Okay. Mm.
yeah.
So we can enter into that topic. Okay. The most important features of DNA molecule are the nucleotide sequence. We know replication, transcription, and translation, and all those things. So the most important features of DNA molecule are the nucleotide sequence and the identification of genes and their activities. Okay. Sandhya, are you
Yes, ma'am.
stable? Okay,
Yeah.
no problem.
No problem.
Okay, any disturbances?
No, ma'am.
Okay, okay. HGP was conceived in 1984 and began in 1990. It's a project, a huge project. Okay, so that started 1984 and almost completed its activities in 1920, but not finished. Primary objective of HGP was to determine the nucleotide sequence for the entire human nuclear genome. We know what is DNA, what is gene, and what is genome. What is genome, Sandhya? What is genome?
Ma'am, the total genes
Hmm.
present in the cellular pool.
Okay, so HGP actually it was the determination of nucleotide sequence for the entire human nuclear genome.
Yes.
HGP was entrusted to elucidate genomes for model organisms like E. coli, yeast, roundworm, and mouse. Okay, at first they conduct HGP for these organisms. Okay,
Yes,
then
ma'am.
it's like an history. Okay,
Uh,
we today's topic is not actually biochemistry. It is like an history and statistics. Like okay,
yes,
okay.
ma'am.
So 26th June 2000, Francis Collins and Craig Winder, leaders of two human genome projects, jointly announced the Human Genome Project and achieved Human Genome Project. Okay. At last, 2000, they announced this Human Genome Project, and the final copy that enters only at 2003. Okay.
Okay.
So the
Yes, ma'am.
work, the starting, the idea comes on 1984 almost. Okay. GP was conceived in 1984 and it is finished only at 26th June 2000 and presented, published at 2003.
Okay. Yes,
Okay.
ma'am.
So these are the leaders of two human genome projects, Francis Collins and Craig Venter. Okay.
Okay.
So the Human Genome Project achievements. What are the Human Genome Project achievements? Actually, the mystery of life is unraveled. We don't know what are the genes, which are the genes present in our body. So
Yes.
with this Human Genome Project, the mystery of life unraveled. The library of life. We have a library of life. Okay.
Yes.
Then, like our chemistry periodic table, we have a periodic table of life.
Yes, ma'am.
Okay, so in case of chemistry, they are all the metals, non-metals, and all type of classification as based on periodic table, right? So in biochemistry, we have a periodic table of life that is known as Human Genome Project.
Yes.
Okay, it's a
Yes,
holy
ma'am.
grail of human genetics, not in biochemistry. Actually, it is in genetics. Okay, so what is the definition? Can you see me that? Can you say the definition of Human Genome Project?
Um,
Actually, I don't given this definition. What is the definition
uh,
according to your idea?
yes,
What
ma'am.
is the definition of? Hmm,
It is to calculate the base pairs
hmm,
that are present in human DNA.
hmm.
Total human DNA, how many base pairs are present?
Okay, I will give you one definition. The Human Genome Project was an international scientific research project with the goal of achieving, with the goal of determining base pairs that make up human DNA. It's a project, a huge project, international project. Almost 20 countries are involved in that Human Genome Project. Okay, France, China, so many countries are there. So then, so this project with what is their goal? goal is determining the base pairs that make up human DNA.
Yes. Yes.
Then they identify and mapping of genes. Okay, identification and mapping of the genes from the physical and functional, from both a physical and functional standpoints. Okay. So we know there are DNA, and DNA made up of nucleotide sequences are there, genes are there. So in this human genome project, we map this. First of all, we speak sequencing and mapping of genes. Okay, according to their physical and functional ways.
Yes, ma'am.
Okay. Okay. So that's about human genome project. And how can we conduct this mapping of human genome? Okay, so the most important objective of Human Genome Project was to construct a series of maps for each chromosome. Okay, we know how many chromosomes are there. Now we are entering into a different step. We are constructing a series of maps for each chromosome. Okay, so the first type of map, that is known as cytogenetic map.
Yes, ma'am.
Okay, what happens in this map? This is a map of the chromosome in which active genes respond to a chemical dye.
Okay. Yes, ma'am.
And display themselves as bands on the chromosome.
Yes, ma'am.
Okay, this is a map of the chromosome in which active genes respond to a chemical dye and display themselves as bands on the chromosome. You can see here, I given here one cytogenetic map. You can see some colorful areas, so darken areas. Yes.
Yes.
Okay.
Yes.
That is why this active genes that respond to a chemical dye.
Yes,
And they display themselves as bands on the chromosome. We know after electrophoresis, we will get some bands, right? In case of protein electrophoresis, alpha 1, alpha 2, beta, gamma, albumin, different bands will be there after electrophoresis. Yes?
Yes. Yes,
when there is a particular M band, what is the disorder? Electrophoresis of protein. We will get an M band in electrophoresis slide. What is the disease? Multiple myeloma. Do you remember? M band will be there, an additional band. Like this, here the dye will create some bands. That bands indicate the presence of active genes.
Yes, ma'am.
Okay, we are mapping the genes according to their physical and functional properties.
Yes, ma'am.
Okay, so when they are active, they will they can be presented with dye. Okay.
Yes.
Then second type of map, that is known as gene linkage map.
Okay.
Okay, a chromosome map in which the active genes are identified by locating closely associated marker genes.
Okay. Yes.
Most commonly used DNA markers are. So what is this gene linkage map? It's also a chromosome map in which we can identify the active genes by locating closely associated marker genes. There will be some marker gene which is closely associated with active genes.
Yes, ma'am.
Okay, how the most commonly used DNA markers, how can we mark this? By 3 or 4 mechanisms are there. It is known as restriction fragment length polymorphism, variable number tandem repeats or mini satellites, short tandem repeats are microsatellites.
Yes.
Okay, do you know these terms?
Only variable number tandem repeats.
Repeats. Okay. Then there are also short tandem repeats are known as microsatellites. Okay. We will discuss it in detail in our recombinant DNA technology. Okay.
Yes.
Then restriction. The third type is known as restriction fragment map. Okay. So in the third one, this consists of random DNA fragments that have been sequenced. Okay, this restriction fragment map means this consists of the random DNA fragments that have been sequenced. Then fourth one is known as physical map. Okay. This is the ultimate map of the chromosome with highest resolution base sequence. We are mainly used to find out the base sequence, base pairs, right?
Yes.
That is
Yes,
the
ma'am.
aim of Human Genome Project. So
Yes.
in physical map, that is the ultimate map of the chromosome with highest resolution base sequence. Physical
Yes.
map that depicts the location of the active genes and the number of bases between the active genes.
Okay.
Okay.
Yes, ma'am.
Physical map. What is the importance of physical map? They depicts location of the active genes and the number of bases between the active genes.
Yes, ma'am.
Okay. So you can see this different 4 maps. Can you see the diagram?
Uh, yes, ma'am.
Okay. Different types of genome maps are there. You can see at first, what is the first type of map?
Cytogen
What is the
genetic map.
importance of cytogenetic map? What is the procedure in cytogenetic map?
Chemical dye that
Dyes
active.
are used. Okay.
Yes. Yes.
The dyes absorbed by active genes or inactive genes?
Uh, they respond to active genes.
Okay. Then what about gene linkage map?
Gene linkage map. Active genes are they're located in associated with marker.
Markers are there. How can we mark this? There are different mechanisms for marker. Which are the mechanisms?
Restriction, fragment length polymorphism, variable number tandem repeats, short tandem repeats.
Okay, very good. That is about gene linkage map. Then third one is restriction fragment map. Yes. What happens
Yes.
in restriction fragments?
They consist of random DNA fragments which are sequenced. Yes.
Oh, yes. DNA undergoes sequencing and they contain a number of fragments. Okay.
Yes.
Restriction fragments are there. We know RE, restriction endonuclease enzyme, and they
Yes.
splits into this, all those things. Okay. So restriction fragment map. Then finally, that fourth map is known as physical map. What is the importance of physical map?
They depict the location of active genes and
Yes.
the number of genes
Base
between the
sequence.
Yes.
Okay.
Yes.
Then finally, we can find out the base sequence with the help of this physical map. Okay. So these are the four important maps. Mapping, gene mapping is this. Okay.
Yes.
Okay. That's about. Then, so you know what is the mapping of human genomes and what are the importance of Human Genome Project? Give me in 4 sentences, what are the importance of Human Genome Project?
The mystery of life is unraveled. It is the periodic table of life. How in chemistry there are many elements periodic table. Like that in periodic table of life, Human Genome Project. Then they are library of life.
Okay. Okay. Then we are entering into genome sequencing. Which are the different types of approaches for? There are 2 different types of approaches for genome sequencing. Okay.
Yes.
Mainly the projects done by 2 research center. Okay. So which are they? Approaches for genome sequencing. Techniques of genome sequencing are useful for the detection of normal and normal and disease gene in humans. It is very important. We can find out which are our normal genes, which are our disease creation genes. Yes? Through this genome sequencing. So mainly 2 approaches were used by 2 HGP groups, Human Genome Project group. They are IHGSC and Celera Genomics. Have you heard about it?
No.
Have you heard about it? IHG, SC and Celera Genomics. These are the 2 centers, okay, research centers. Okay. So, IHG-SC employed map first and sequence later. Okay. IHG-SC employed map first and sequence later. The principal method was hierarchical shotgun sequencing. Which method they used for sequencing? Okay. IHG-SC employed map first and sequence later.
Yes, ma'am.
Okay. The principal method was hierarchical shotgun sequencing. Okay.
Yes, ma'am.
That is the method they used for sequencing. This technique that involves fragmentation of genomes into small fragments. Okay. Then inserting them into vectors. You know
Okay.
vector, which are known as vector.
Uh, they they are like circular DNA. They have it is and they incorporate the desired gene into the DNA.
Okay, very good. So inserting them into vectors and finally cloning. Okay.
Yes.
Then these cloned fragments can be sequenced.
Yes, ma'am.
Okay. So that is the method by IHGSC. Okay. So employed map first and sequence later. The principal method was hierarchical shotgun sequencing. This technique involves fragmentation of the genome into small fragments, inserting them into vectors and cloning. Cloned fragments can be sequenced.
Yes,
Okay, Sandhya? Yes. Then what happens in Celera genomics? In Celera, they use whole genome shotgun approach. Okay. They
yes,
first of all, they bypasses mapping step and saves time. Okay.
yes,
And the Celera group have high throughput sequenators and powerful computer programs for the completion of human genome sequence. So Celera is more advanced, more easy project. Okay, they use
yes,
whole genome shotgun approach, mapping step is avoided and they saves time, and they have high throughput sequenators and powerful computer programs for the completion of human genome sequence.
yes.
Okay, then next, uh, then what is your idea about the two approaches for gene sequencing and its importance?
IHGSC, it is they first they employ map and then they sequence. The method used is hierarchical shotgun sequencing. In that method, the genome is fragmented into small fragments that are inserted into vectors, mostly bacterial artificial chromosomes, and then cloning can be sequenced. The second one is Celera genomics. The whole genome shotgun is taken. So by passing mapping step, the time is saved. They have through from that last point. I didn't understand that Celera.
Okay. Celera, they use some special equipments. Okay, they are known as throughput sequenators. Okay, for clarity in the human genome project is done by this throughput sequenators and powerful computer programs. Okay, that's all.
Yeah.
That's all. Okay, then next we are entering into whose genome was sequenced. Okay.
Yes.
So many individuals are there, then which one's genome was sequenced?
Yes.
It's a question. Yes? So it does not matter whose genome is sequenced, since there is phenotypic differences between individuals are due to variations in just 0.1% of the total genome sequence.
Okay.
Yes? Generally, we have so many differences.
Yes, ma'am.
We know phenotypic variations and genotypic variation. So the phenotypic differences between the individuals are due to variations in just 0.1% of the total genome sequence.
Yes.
Okay. Therefore, many individual genomes can be used as a source material for sequencing. There is no peculiarities for the selection whose genome was sequenced. The reason is that there is only 0.1% of phenotypic difference.
Yes.
Okay. Most of the genome sequences identified. About 90% of the human genomes are sequenced. Okay.
Yes.
It is composed of 3.2 billion base pairs. Wow.
Yes.
Yes. We can't find it out. So many examples are the 3.2 billion base pairs. Yes. Okay. Then if the genome is recited at the rate of 1 base per second for 24 hours a day, it would take a century to recite the book of life.
Yes.
Okay. So if we written in the format of a telephone book, The base sequence of human genome would fill about 200 telephone books of 1,000 pages each.
Okay. Yes,
It's a
ma'am.
huge project. It's a heavy project. Okay.
Yes.
That's why our medical subject, the last word is end with God.
Yes.
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 No science. Our final word is God.
Uh,
Yes, this much content. How he arranged in a
mm,
perfect manner. Yes.
yes, ma'am.
There is molecular biology, genetics, and everything, but no one can arrange this base sequence and all those things this much perfect way.
Yes.
Okay. By human effort, that genome sequenced and it is composed of by human effort. We conduct one research project, and through this human genome project, we sequenced our human genome, and it is composed of 3.2 billion base pairs. Okay,
Yes.
it will take a lot century to recite the book of life.
Yes, ma'am.
Okay, okay, that's the importance. Then individual differences in genomes. There may be some individual differences in genome. Every individual, except identical twins, have
Ah,
their own versions of genome sequences.
okay. Yes, ma'am.
The differences between individuals are largely due to a main thing that is known as single nucleotide polymorphism.
Yes, ma'am.
Okay, it is known as SNP. What is SNP? SNP represent position in the genome where some individuals have one nucleotide and others have another one.
Okay. Okay. Yes, ma'am.
So like our point mutation.
Yes,
Okay,
ma'am.
addition or deletion. Purine to purine.
Very much. Transmission.
Purine to purine. Here it is A replaced by G.
Yes,
Adenine
ma'am.
replaced by guanine. So the
Yeah.
frequency of occurrence of SNP is estimated to be 1 per 1000 base pairs.
Yes, ma'am.
Okay, this is the only difference in individual differences in genomes. Okay, every individual except identical twins have their own versions of genome sequencing.
Yes, ma'am.
The differences between individuals are largely due to single nucleotide polymorphism. SNPs represent position in the genome where some individuals have one nucleotide and others have the other one. Okay, the frequency of occurrence of SNPs is estimated to be 1 per 1000 base pairs. Right?
Yes. Yes,
Okay.
ma'am.
Then organization of human genome. How can we have a 3.2 billion base pairs? Okay, we
Yes.
total genome project contains or we sequence almost 90% of genome, then we get almost 3.2 billion base pairs. So how can we organize this human genome? The total human genome is 3200 MB. Okay,
Okay. Yes,
only
ma'am.
a small fraction that represents the actual genes. Okay.
Yes. Yes,
Then,
ma'am.
while the rest is due to the gene-related sequence are introns, pseudo genes. We discuss about pseudo genes. Yes.
Yes. Yes, ma'am.
Yes. At the time of regulation of gene expression, we already discussed there is pseudo
Yeah.
genes.
Yes.
Junk DNA, yes. Then intergenic DNA, which are known as intergenic DNA, long interspersed nuclear elements, short interspersed nuclear element, microsatellites, DNA transposon, so many other things are there.
Yes, ma'am.
Okay. So the intergenic DNA that represents the parts of the genome lies between the genes which have no function, which is regarded as junk DNA.
Yes, ma'am.
So, in short, there are two regions in human genome. That is, one area is known as junk DNA.
Yes, ma'am.
That is inactive forms of genes. Another area, it is known as active forms of genes. Clear?
Yes, ma'am.
That's all. So that's about the organization of human genome. You can see here, yes?
Yes. Yes, ma'am.
You can see here, this
That.
human genome. Hmm?
Yes, ma'am.
Yes, Cynthia?
Yes,
Can
ma'am.
you see the slide? Yes?
Yeah. Yes, ma'am.
So human genome, genes and related gene sequences, that's about only 1200 MB. Okay. Then Sandhya, hello.
Yes. Yes.
Okay. Then the next part is known as intergenic DNA that contain long interspaced nuclear elements, short interspaced nuclear element, okay, LTR elements and DNA transposons, microsatellites and other. Total 2000 MB is known as intergenic DNA or junk DNA.
Okay. Yes, ma'am.
Okay, actually, it is total amount is 3200 MB human genome, and this 1200 MB is the active form of genes, and intergenic DNA that's about 2000 MB.
Yes.
Okay, then next, what is the individual differences in genomes?
Yes, ma'am. This um, the except twins, they variation there is they have their own version of genomic sequence. That is difference between individual is because of that single nucleotide polymorphisms. It's like point mutation. They represent position in genome where individual of one nucleotide is with others. Like individuals differ from that single.
Then organization of human genome. Can you explain the organization of human genome?
Yes, ma'am. The total human genome there is 3200 MB. Um, um, that is divided into genes that are 1200 MB and 2000 MB. The 1200 MB is genes and related gene sequences, whereas 2000 MB is intergenic DNA. Intergenic DNA that represents parts of genome that are present between the genes but have no function, like junk DNA.
Okay. Then next is genes present in human genome. Okay. Before the result of FGP, best guess of human genes are almost 80,000 to 100,000, 1 lakh. Okay. So this is based on the fact that the number of proteins in human cells is equal to 1 lakh. So many genes are expected. But actually the number of proteins is almost is equal to 1 lakh. But here the number of genes are lower than the proteins. Why this number of genes are lower than the proteins? What is the reason? The reason is RNA processing. We know splicing. What is splicing, Sandhya?
Ma'am, the splicing like that in the DNA, the removing of non-coding
in the DNA or RNA? In the DNA or RNA?
RNA. We remove the introns and
yes, that there are some coding sequences and non-coding sequences, right?
Yes.
We remove that non-coding sequences at the time of which time RNA processing, post-transcriptional modifications. Yes, we arrange that RNA and we rearrange that RNA with an active form and convert it into an active form before enter into translation. So that particular process is known as RNA processing. Yes, at that time some amount of genes are cut, right? Which one, exons or introns?
Introns are cut.
Yes. So inactive form or the non-coding sequences are removed. Only the coding sequences are ligated, right? Joined together. So there is a decrease in the number of genes. So number of proteins will be greater than the number of genes. Okay. So we expect almost 80,000 to 100,000 genes are there. First human guess. Because we have 80,000 to 100,000 proteins. So we compare equal to protein, there may be genes. But it is wrong because the number of protein is greater than the number of genes. Because at the time of RNA processing, okay, in the time of splicing, we remove that intron portion.
Yes, sir.
Okay, so the number of genes are lower than the protein because of RNA processing. Single mRNA can code for more than one protein.
Yes,
that is also an important point. Single mRNA can code for more than one protein. Okay, then A broad catalogue of human genome in a pie chart. Almost 17.5% of gene that participate in general biochemical reaction, 23% in the maintenance of genome, then 21% that is used for signal transduction, and 38% involved in the production of proteins. That is a broad catalogue of human genome. Yes? Okay.
Yes.
Okay, Sandhya. What are the roles of different genes? That's all. Okay. 17.5% general biochemical reactions, 23% maintenance of genome, 21% signal transduction, 38% in the production of protein.
Yes.
Okay.